We're going to do a quick review of protein metabolism. So when you eat proteins, you break proteins down into amino acids. So once I have the amino acid, I'm ready to do transamination. So that's the first process we're going to talk about. So I have my amino acid. Oops. And what I'm going to do is I'm going to take the amino group off of it. So I'm going to take the amino group off. So if I remove the amino group, I end up with a keto acid, also known as a carbon skeleton. So be comfortable with the term carbon skeleton. A carbon skeleton is an amino acid without the amino group. So it's actually a keto acid. Transamination, though, is kind of like oxidation and reduction in the sense that I can't, I can't oxidize something without reducing something. I can't reduce something without oxidizing something. I can't take an electron off and just leave it there, or I can't expect to have electrons floating around that I can just grab one when I need it. So I can't take an amino group off and just leave it there. I've got to put it on something. The something that I put it on is alpha ketoglutarate. So look at alpha ketoglutarate as your amino group holder. When alpha ketoglutarate is holding the amino group, it becomes glutamate. So it's like it's the amino group holder. When it's not holding the amino group, its name is alpha ketoglutarate. When it's holding the amino group, its name is glutamate. So I have that amino group now off of the amino acid. I can take the keto acid or the carbon skeleton, go and make energy out of it. That's how I get energy from my proteins. And remember what we said about that. Depending on what the amino acid is, that determines what I'm going to do with the keto acid or the carbon skeleton. The two that we talk about the most are this. For example, if my amino group is aspartate, or my amino acid is aspartate, if I take the amino group off of that, I'm going to get oxaloacetate. So if I asked you right now, what will you do with oxaloacetate? You'll say, well, I could put it in the Krebs cycle, maybe gluconeogenesis. Of course, it depends on what the cell has and what the cell needs, but you know all the places you can put oxaloacetate. Or the other one we talk about a lot is alanine. If I take the amino group off of alanine, that's going to give me pyruvate. So what do I do with pyruvate? Well, of course, it's one of those it depends answers. So do I need glucose? Well, I do gluconeogenesis. Do I need energy? Well, I can put it in the pyruvate dehydrogenase complex, the Krebs cycle, and so on. So my point is you already know what to do with that pyruvate, how to get energy or whatever you need out of it. Now we've just got to deal with the glutamate, that molecule that's now carrying that extra nitrogen. Because remember, we have no storage molecule whose purpose is to store nitrogen, store amino acids, or store anything as fuel. You're full of proteins, you have all kinds of protein in you, but the primary purpose of all of those proteins is always going to be something other than fuel storage. If you break a protein down for fuel, like a muscle tissue or whatever, that's not what it was meant for. So keep that in mind with application-based questions. For transamination, our enzyme that does that is going to be an aminotransferase, also known as a transaminase. And the first part of the word will be the name of the amino acid. So I have my glutamate now. I've got to do something with my glutamate. So we're going to do that next. So I have my glutamate. And what I'm going to do is oxidative deamination. So really pay attention to the name of the process. It's telling you what's going on. The enzyme I'm going to use for this is glutamate dehydrogenase. So here's what I'm going to do. I'm going to take the glutamate, I'm going to oxidize it back to alpha ketoglutarate. While I deaminate it, take that free ammonia off. And while I'm oxidizing, remember I always have to reduce. I can't oxidize without reducing. I can't reduce without oxidizing. So I'm going to reduce an NAD to an NADH. Now I have this free ammonia. I can take the free ammonia, send it to the urea cycle, and get rid of it. So what have I done? What I've done in the name of metabolism is this. I've done transamination, followed by oxidative deamination, followed by the urea cycle. So that's my order of operations for metabolism. Of course, transamination gave me the molecule that's going to go and give me the energy from the amino acid. The rest of these steps are just about dealing with that ammonia or that nitrogen I can't allow to stay in my tissues. So let's go back though. Remember with transamination, if it's going to let me draw on it, with transamination, there's something really important about transamination. Transamination is a reversible process. So going down the page, the way we have it drawn here, that's about metabolism. However, we can go up the page, and we talked about this, so let me remind you. If I'm going up the page, as in turn these arrows around, that's going to be about making non-essential amino acids. So let's redraw that going the other direction. So if I want to make non-essential amino acids, and let me remind you what essential means. An essential amino acid is one I need to eat. The only way I'm getting it is eating it. That's the essential part of it. It's essential that I eat it because I can't make it. So I can only make non-essential amino acids. So to make non-essential amino acids, I need a couple things. So if I want to make that non-essential amino acid, I need two things. I need the carbon skeleton, and I'll get that somewhere in my metabolism, in my reactions. So I need the appropriate carbon skeleton for whatever amino acid I'm making. If I'm making aspartate, the carbon skeleton I need is oxaloacetate. If I'm making alanine, the carbon skeleton I need is pyruvate, and so on. I also need a molecule of glutamate, because what I'm going to do is this. I'm going to take the amino group off of the glutamate. So if I take the amino group off, that gives me alpha ketoglutarate back. I just go back and forth, alpha ketoglutarate, glutamate, depending on whether I'm putting an amino group on or off. When I take it off because I want to put it on the carbon skeleton and make my non-essential amino acid. So carbon skeleton, I get somewhere in my metabolism. But where do I get the glutamate? Where I get the glutamate is another process that's awfully similar to oxidative deamination. I'm going to do a process called reductive amination. With these, I don't remember when we were doing it in class if I was drawing up the page or down the page. Really doesn't matter, but I'm pointing it out because I might be doing it opposite to what we did in class. So I want to make a molecule of glutamate. I'm going to do that by reducing and aminating alpha-ketoglutarate. So I'll put a free ammonia in, but since it's a reduction, I need to oxidize something. I'm going to oxidize NADPH to NADP. The enzyme I'm going to use, again, is glutamate dehydrogenase. So that gave me the glutamate to plug into transamination and make my non-essential amino acid. So if my mission is to make non-essential amino acids, what I'm going to do is reductive amination, followed by transamination. Here's where transamination kind of catches you a little bit with questions. We've been talking about and focusing on metabolism since the course began. We're all about how are we getting energy for our cells from the food, the fuel that we eat. So you definitely have metabolism on the brain. Because of that, you think of transamination as the first step in metabolizing your proteins. You're absolutely right on that. However, the problem is when you're thinking that, what you're forgetting is that transamination is a reversible process, and the reversible process makes you your non-essential amino acids. So it's not an uncommon thing to get asked, which of the following processes makes me my non-essential amino acids? If you get asked that, you're going to pick transamination. Reductive amination makes me the stuff that makes me non-essential amino acids. The stuff I'm referring to being glutamate, kind of like the Krebs cycle makes me the stuff that makes me ATP but doesn't make me ATP. Reductive amination makes me the stuff, glutamate, that makes me non-essential amino acids, but it does not make me the non-essentials. Transamination actually makes me my non-essential amino acids. So be careful there. Also, be prepared for those questions. That's the list of processes. It'd be something like transamination, oxidative deamination, and we've done these in class, reductive amination, and probably the urea cycle. And the question would be something like, you ate a lot of protein. Which one of these would you not expect to be doing as a result of eating a lot of protein? So, of course, if I ate a lot of protein, I'm not going to be doing reductive amination, things like that. Or here's your list. Which one of these makes me non-essential amino acids? Of course, transamination is what you would pick there. So be prepared for that kind of question. Also, be prepared for somebody playing games with this. Let's compare oxidative deamination to reductive amination. So in oxidative deamination, I took glutamate to alpha-ketoglutarate, took a free ammonia off, and reduced NAD to NADH. In reductive amination, I took alpha-ketoglutarate back to glutamate. I put a free ammonia in there, sounding awfully similar, except I oxidized NADPH to NADP. So all I've got to do is tell you, well, it's the same enzyme going both directions. So it's the same enzyme. Doesn't that make these reversible reactions, the reverse of each other? No, they're not. To be a reversible reaction, everything's got to be the same. If I change one thing, that makes it a different reaction. So look at the one thing I changed. I have in oxidative deamination NAD going to NADH. In reductive amination, I have NADPH going to NADP. So here's a really common type of question to get. We just did the application type of thing where, you ate a lot of protein. What will you do? What will you not do? What process makes non-essential amino acids? Don't forget the nitty-gritty questions that everybody likes asking you. All of the following are products of one of these or the other except. So let's look at that. So first, let's do the products of oxidative deamination. So the products of oxidative deamination, NADH, free ammonia, and alpha ketoglutarate. So they're my products of oxidative deamination. What are my reactants of oxidative deamination? My reactants are glutamate and NAD. Reductive amination. What are my products? My products are NADP and alpha ketoglutarate. What are my reactants? Glutamate, NADPH, ammonia. So what really everybody will be looking for there is two things when we give you this kind of question. One is, do you know what's going on with your NAD stuff? Which one goes where and which direction are you going? The other one is, do you know what's going on with your direction with alpha ketoglutarate and glutamate? So make sure you're really solid with the comparison. They're not reversible. We have the difference with the NAD stuff, and I'm saying NAD stuff on purpose. NAD going to NADH in oxidative deamination. NADP going to NADPH in reductive amination. Also, which way am I going with glutamate and alpha ketoglutarate? So those questions, like I said, could be application based or could be nitty gritty like that. Okay, the other thing that questions are very predictable on and expect questions on it, of course, is the urea cycle. So the urea cycle, because we've done so many processes, you should be just ready to hammer out, well, what kind of thing do I expect to get asked? And if, as long as you can answer that, you're going to be correct. So the urea cycle is a little bit different in the sense that part of it takes place in one part of the cell, part of it in another. I'm going to start out in the mitochondria with the urea cycle. I'm going to start out with my free ammonia. I'm going to start out with a carbon dioxide, and I'm going to start with 2 ATP. Of course, we have ATP stuff going on. Those molecules are going to react and form a carbamoyl phosphate. Using carbamoyl phosphate synthetase, the regulatory enzyme. I do that in the mitochondria. Then I go and find myself this molecule that's both a product and a reactant of the urea cycle. Which of the following molecules is both a product and a reactant of the urea cycle? Ornithine. So I made ornithine as the product of the last round of the urea cycle. I use it in the first reaction of my next round of the urea cycle. I've got that product reactant thing going on, and that's what makes it a cycle. I've got to have something close the loop or it's not a loop, and not a loop means not a cycle. So I made it in the last round. So that's my product and my reactant. However, I made it in the cytoplasm. So it has to cross into the mitochondria to react in that first reaction. So which of the following molecules crosses from the from the cytoplasm into the mitochondria? It's going to be your ornithine. So my ornithine reacts with my carbamoyl phosphate, makes me a molecule of citrulline. So then my citrulline has to cross out to continue in the urea cycle. Which of the following molecules crosses from the mitochondria to the cytoplasm in the urea cycle? Of course, that's going to be your citrulline. So urea has two nitrogens in it. We've accounted for one. We have one from this free ammonia. So I have to have a source of the other nitrogen. So the molecule that brings the second nitrogen in the urea into the urea cycle is aspartate. So this is one of our plan A, plan B strategies with a question. Plan A, if you're asked where the second nitrogen comes from, plan A is aspartate. That's the molecule that actually enters the urea cycle carrying that ammonia. However, if and only if you're not offered aspartate, you're going to pick glutamate. I won't do this to you, but if you get this question somewhere else, you're going to pick glutamate. Because what happens is that nitrogen in the form of an amino group is on glutamate. I transaminate glutamate, meaning I take the amino group off of glutamate, gives me alpha ketoglutarate back, put it onto oxaloacetate, that gives me my aspartate. So the last stop for that amino group was glutamate. So if somebody else asks you the question and doesn't offer you aspartate, if I ask you, I'm offering you aspartate. If somebody else doesn't offer it, plan B is your glutamate. So my aspartate comes in, reacts with my citrulline, forms me argininosuccinate. Okay. But forms me argininosuccinate and uses an ATP. So I use another ATP here. One ATP gets used. And that's the last ATP I'm going to use. So the total of ATPs I use is 3. So how many ATPs does it cost to make a molecule of urea in the urea cycle? It costs me 3. However, there's an oddball thing here that we do just here, not other places. What color haven't I used? Haven't used red. This ATP goes to AMP. So in the process of going from ATP to AMP, I made 2 phosphates. Over here, 2 phosphates going to, or I'm sorry, 2 ATP going to 2 ADP gave me 2 more phosphates. So the number of phosphates I make is 4. So be careful with that question. Is the question asking you how many ATPs did you use? Your answer will be 3. Or is the question asking you how many phosphates did you make? Your answer is going to be 4. Here's where the question gets messy though, and it's a reading thing. I can start out in that question talking about I used ATP. So I used ATP, and in the process of using ATP, I made how many phosphates. So be careful. Is the question asking how many ATPs did you use? Or is the question asking how many phosphates did you get? So I get the argininosuccinate, I cleave it. Part of it goes to fumarate, which cycles back around to oxaloacetate. Fumarate to malate to oxaloacetate for my office. Other part becomes arginine. I cleave the arginine into my urea. My urea then can go to the kidney, get incorporated into the urine, and get gone. The other part of the arginine goes to ornithine and starts all over again. So that's it for your urea cycle. Be careful also with some combined questions. So a nice combined question like we talked about would be something like this. I could have my ornithine, my citrulline, my oxaloacetate, my citrate. And my question would be very simple. Which one of these cannot cross the inner mitochondrial membrane. And when you get a question like that, think about, where have I seen these? So do the where have I seen these, and that'll help you. Well, where did I see ornithine? That was in the urea cycle, and what was it doing? It was crossing from the cytoplasm into the mitochondria. Not it. Get rid of it. Citrulline, where did I see it? In the urea cycle. What was it doing? Crossing from the mitochondria to the cytoplasm. Not it, because it's a cannot question. Oxaloacetate, where did I see that? Well, I saw it in the Krebs cycle. What was it doing there? Just recycling, so nothing about crossing. Where else did I see it? I saw it in an electron transport shuttle. What was it doing there? Well, what it was doing there really was getting stuck, not crossing. Then that's my one that can't cross. Or where else did I see it? Gluconeogenesis. What was it doing? Well, if I'm in the cytoplasm, it's just going to keep on trucking to make me glucose. Or if I'm in the mitochondria, it's not crossing. It's got to go to malate to cross. So again, there's confirmation. It's my answer. Citrate, where did I see that? Let's make sure we have the right answer. Well, the Krebs cycle, what was it doing? Well, just doing the Krebs cycle. All right, not helping me there. Where else did I see it? In fats. What was it doing there? It was crossing. So it's not my answer. But look at all the categories in a rubric we can check off. It's a protein question. So it's a protein question. Gluconeogenesis. It's a carb question. It's a fat question. So in a rubric, it could be any of those. So that's why we're spending the time on questions like that. I think you're going to get a lot of mixed questions like that. So anyway, that was our quick review of proteins, protein metabolism.